First one.
Yeah.
It's your first time presenting here?
No,
No, you did present
maybe my
before.
second.
Second time? Okay. You know, it works, right? Up and down, left and right. All right. I may switch this to that side. Right now it's video conference to the Jewish general, and usually they show a few people in each room.
Right. Yeah.
I think today's an exam day, so it's gonna be less.
Fine by me.
Go.
So I hear your company got, I get all the audience follow-ups all the time from Mark. Oh yeah, Mark. He talks a lot. Protocols as well. You got a letter and all kinds of stuff. I was here. Oh yeah. Makes sense from what you're about to say.
Reminders of payment.
At the end, well, I guess the event numbers, but
Lo de atrás, lo de atrás es el completador. No ya mis
it's kind of late from the snow, I think.
Well, Dr. Gomez, is it possible to mute your microphone, please?
Oye, esto,
Hey, how are you? You too. Oh, right, okay. Good morning, everybody. First, Happy New Year to everyone. I wish you health and success in everything that you undertake. So it's really my pleasure this morning to first congratulate everybody who braved the weather this morning to come in and introduce Joshua Sonnen, who is a neuropathologist who recently joined the Neuro. He's been in town for about 6 months, and he's setting up here at the Neuro, and I'm really hopeful that we're going to have very meaningful interactions with him in the near future. So Dr. Sonnen trained at the University of Washington, working with the ADRC cohort, and he is very much interested in, of course, the molecular underpinnings of age-related disorders, and he's going to talk to us about this. I think it's very important to really think about these things more and more because of the direction that we're taking for the treatment of neurodegenerative illnesses. Molecular diagnosis is going to become something very important, I believe, and I think we're going to get interesting insights from his work and his ideas about how to achieve these things. So welcome, Dr. Sonnen.
Thank you. So it's nice to see everybody this morning. So first of all, I have no conflicts of interest, and I'm going to talk about some things that are a tiny bit controversial in the pathology world, and these are just my opinions, they do not represent the opinions of anyone else. And I'm going to talk about some limitations of certain types of data, but just for fairness sake, all cohorts have their own inherent limitations, and there's no real perfect one. And although the title, I like to think that I view everything through sort of an epidemiologic lens because I've worked with population-based cohorts. Population-based autopsy cohorts are not really truly epidemiologic, and so they have their own limitations. So I presented here a couple of years ago, and I presented a talk that had a similar title, and at that time I presented a review of all of the work that I had done and tried to neatly tie up everything in a bow. And this time it's going to be a little bit more messy, but I've tried to include information at a variety of levels, including a fair amount of didactic information about the neuropathology, specifically underlying cognitive impairment in the age-related setting. The reason that things I think are going to be messy, I've moved from, we're going to be moving from the world of eponyms, which are, concise names for diagnoses, and in the last couple of decades, I think there's been a big move away from eponyms to descriptive names of disorders, which in general I, support. The problem being that our descriptive names have gotten longer and longer, and as such, we've moved to ever longer acronyms. So the second half of my talk is going to be laden with a variety of neologisms and acronyms that are a little bit unwieldy. And I'm uncertain whether or not they're going to stand the test of time or not. I'm also going to move from, previously I talked about data that was really based on observational autopsy data in an epidemiologic setting, and in that lens I feel that I have some insight into what's actually going on in the human brain, and instead we're now moving, I'll be presenting more expert consensus information, which is not as data driven. So I'm less certain how to interpret that, and I put the asterisks in saying, special concern for statistical significance. Actually, most of the expert consensus papers that I've looked at don't really have much in the way of statistical testing, and when they do, it tends to be sort of circular. Probably the highest quality data is looking at inter-observer variability, which has really the same has the same outcome as your test is the same outcome as what you're testing. I'm a neuropathologist. Inherently, everything that I'm going to do is going to be descriptive. The value that the main value that I think that a neuropathologist can add is we can offer insight into relevance to human disease. When we work with in animal models, we're always looking towards how does the animal replicate what is happening in human disease, and trying to use information from human cohorts to try to offer us some insight into underlying prevalences and mechanisms. I don't know that we'll ever really that neuropathologists in our work ever really prove anything new. We don't have nice controlled studies to do that, but probably the highest level of data that or the highest level of proof that we get is the ability to identify something in one cohort and then replicate it in other cohorts. And I'll have an example of that later on. The types of data that I've enjoyed working with are those that include multiple different characteristics. Traditionally, this type of neuropathology data has been gotten from a variety of different sources. Getting neuropathologic data, usually requires autopsy, which usually limits you to samples of convenience. The strength of those tend to be that diagnosis is considered the gold standard, which is, at least it's cross-sectional data and it doesn't change over time. So we can at least see what's happening inside people's brains. It does tend to be overly reliant on expert criteria, and there are questions as to whether or not it can be generalizable. Of course, epidemiologic cohorts give us the benefit of finding clinical diagnosis from populations that are inherently relevant to human disease. But because epidemiologic cohorts generally have not had autopsy data, clinical diagnoses often do not match pathological diagnosis. Even in the best cohorts, 80% concordance is generally considered excellent. And there's, especially in these cohorts, there's an awful lot of comorbidity. And in the past, I think this has led to a great deal of confusion. Later on, I'll get up on my soapbox a little bit and reiterate a short part of my prior talk, just to try to highlight what I think the importance of autopsy data is. And then there are focused research cohorts like the Alzheimer's disease research centers where I trained that are able to pair neuropathology data with longitudinal clinical data. And the main limitation of these cohorts are that they are highly selected. And in the past, highly selected has meant that they've accumulated Alzheimer's type pathology at the expense of all other pathologies. And that tends to be especially relevant in dementing diseases where, as I said before, comorbid disease is very common. And comorbid disease, especially in the world of vascular injury, tends to be, I think, at least my opinion is that it is etiologically distinct from Alzheimer's type pathology, has different risk factors, probably has different mechanisms, and likely has different treatments. And in the past, this will get back up on my soapbox, there's been confusion that has caused, I think, a great deal of investigation, perhaps in the wrong directions, in my opinion. So the first half of my talk, I'm going to talk about some of the insights that have been gained from some of these, a small number of longitudinal population brain autopsy cohorts. Here I'm just referencing some of the earliest of these studies. Since this time, I think you could reasonably add another 4 or 5 studies that would fulfill all of these criterias to some level of stringency or another. One of the earliest was the nun study, a cohort of almost 150 Carmelite nuns who were ordered by their Mother Superior to donate their brains and offered us some of the earliest insight into the prevalence of Alzheimer's type neuropathology and were some of the earliest clinical pathological samples that were able to be done in a population-based or epidemiologic sense. I'm going to then talk about the Honolulu Asia Aging Study and the Adult Changes in Thought Study, which are two studies that I've worked with extensively, and from which I think we've learned, at least personally, a way to conceptualize what the common neuropathologies underlying dementing illness are. And then I'll talk about the Religious Order Study and Memory and Aging Project, which which are a group of sister studies that I think are sort of the premier longitudinal population-based autopsy studies in existence. So the Honolulu Asia Aging Study, these are decedents from the Honolulu Heart Program, which was a part of a three-part population-based study looking at ethnically Japanese men in Honolulu. It was part of a larger study, the Nihonsa, the study that compared them to Japanese in Japan, Japanese men in the US, and then in Hawaii, looking at sort of three different types of acculturation and cultural exposure. It's supposed to be a genetically homogeneous group of individuals that then had exposure to either Western or Japanese foods and other cultural exposures. But we're not really talking about that. A very influential study that taught us a great deal about the pathology underlying cardiovascular disease and stroke. But individuals in this study, it accounted for almost all of the Japanese males on Oahu, and in a 20-year cohort, and followed them throughout life, throughout midlife into late life, as they developed cognitive impairment. It was very influential, one, because it was one of the earlier studies that could be considered an epidemiologic neuropathology cohort, but also because it was agnostic to the causes of dementing illness, and basically attempted to look at, had a blinded neuropathologist look at essentially any pathology that they could find in brain, and then looked at them as predictors in a combined model for dementing illness. We attempted to replicate the findings then in the Adult Changes in Thought study. This is an ongoing longitudinal study that has been going on in King County in Washington State for about 20 years before I did my training. And it was a random sample of 3,400 individuals who were all part of a single managed healthcare concern. They all had similar access to medical care. They had a relatively high rate of autopsy, about 20%, and extensive drug exposure data. Whereas the Honolulu Asia Aging Study accounted for the majority of individuals in the population, the Adult Changes in Thought study had this 20% autopsy rate. And although the general cohort was considered to be representative of the population in general, there was a pretty significant concern for selection bias in that smaller population, something that we did in a lot of our study, and actually every subsequent study in this cohort was try to account for selection bias using models that basically use inverse probability weighting. So basically you more heavily weight individuals that are underselected in the population to try to get an estimate. In the end, the characteristic of the cohort that tended to be most predictive about whether or not someone died and came to autopsy was whether or not they were demented. Our working underlying theory is that people, at least in King County, they require sort of this dual consent process where people have to consent to an autopsy before death, and then their families are reconsented after their death. And so what we think happens is that after people die, if they were demented, their families are more interested in receiving an autopsy, and so they were about twice as likely to come to autopsies. In the end, this didn't change major associations with outcomes, but it was comforting to know that we were able to at least investigate this. And then the third study that I'll talk a little bit about is the religious order study. Also the religious order study and the Memory and Aging Project. So the religious order study began in 1994. It was a cohort, or is a ongoing cohort of nuns and priests, Catholic priests in the U.S. With 3,400 individuals originally enrolled, about 10% minority. The vast majority of these individuals who die come to autopsy. 88% autopsy rate is excellent. And because of this high rate, they've resisted being concerned about selection bias. I don't know if that's a mistake or not. I suppose time will tell. But this really is one of the premier studies. They do extensive neuropathology on all of their cases. The pathology is of very high quality. It's really quantitative neuropathology, whereas the majority of other studies tend to do more semi-quantitative evaluations. But here, especially with Alzheimer's type pathology, they look at random fields, they do random counts, do averages. That's not really how most neuropathology data has been done. And I'll go into some of the specifics of what those data are, and what their limitations might be. One of the critiques of this cohort was this 10% minority population, which is lower than the U.S. minority population by a factor of about 3.5. The Memory and Aging Project was a second project that was at least partially designed to get a more diverse to bring a more diverse population into investigation. It's general population from Northwest Illinois. Both of these are centered in Chicago at Rush University. And they attempted to oversample for minorities with some levels of success. Over the years, this is one of these two sister studies are, I think, the best funded epidemiologic or community-based neuropathology samples in existence, and have continually added layers and layers of proteomic, genetics, epigenetic data over time. This is just, sorry, it's a little blurry, but these are just some of the different types of data that have been collected. I'm not going to talk about all of those. I just wanted to highlight the incredible power of this study. Okay, so what have we learned from some of these studies, or what are the take-home messages? And from the neuropathology side, in terms of just cognitive impairment, there are 3 common causes of dementing illness. So what do I mean by common causes? I'd say, over individual causes of dementia that account for more than maybe 1 or 2% of individual dementia diagnoses, at least from a population-based setting like this. And not surprising, the largest one is Alzheimer's disease. And I'm going to get into the nitty-gritty of exactly how Alzheimer's disease is evaluated in the neuropathology setting, how that's evolved. It's complicated. The second is vascular disease. In the first two cohorts, at least, the pathology that we've used to assess vascular disease has been cerebral microinfarcts, and this has worked great. This was discovered in the Honolulu Asia aging study. We confirmed it then in the ACT study. Yes? So these were all put into a single regression model. And so we looked for independent predictive power of individual pathologies. And when we did this, the individual pathologies that accounted for dementing illness, or accounted for cognitive impairment, or diagnosis of dementia, actually they both ended up being pretty similar, were Braak stage for Alzheimer's disease, cerebral microinfarcts, and the presence of neocortical Lewy bodies. And at least in the ACT cohort, they all had a relative risk of about 5 times. So you're about 5 times as likely to be demented if you had high levels of these pathology as if you had no pathology. But very specifically, these were not binned into, AD plus vascular disease only, AD plus Lewy body disease. I'll go through some of those bins later in just a second. But these are just, I'm just interested in what are the general, what are the independent contributions so that I can get an idea of, and these are, I think statistically, at least Alzheimer's disease and vascular disease appear to be statistically independent.
We have control of this? Because they're all, by definition, they're all dementia, right?
No. This is a population-based study, so about a third to a sixth have significant cognitive impairment at the time of death. So there's no separate control population. It's really an epidemiologic study. That being said, there are, and I'll talk about this in a second, there are people that have high levels of pathology who are not demented. There are people that have low levels of pathology, that not very many, that are demented. So these are general risks. Part of that, I think, has to do with limitations of measurement. When we're making these estimates of pathology, we're sampling small areas of the brain. I don't know what we don't know what the true absolute burden on the brain is. That I think imaging studies probably offer us more insight into global brain injury. Okay, as I said, comorbidity is common in aging. So if we took those 3 common causes of dementing illness and put them in a population in general, no pathology. So here's the x-axis really doesn't mean anything. We've just ordered all individuals in. We've ordered individuals in cohorts as to the level of pathology they have. Here, blue is Braak stage for Alzheimer's pathology, and separated into 3 groups. The blue can be either, no Alzheimer's pathology, low, intermediate, or mild, moderate, severe, and then red is vascular disease, and blue is Lewy body disease. And these are combined pathologies in 4 different population-based cohorts. And here we're actually looking at people that have no dementing illness. So there's an awful lot of comorbid pathology out in the general population, even in individuals who are not known to be demented at the time of death. The rates of comorbidity when we originally did this, I thought that all of the pathologies were likely statistically independent. The rates of comorbidity appeared to be at the rate that you would expect by chance alone. So if, let's say, 50% of our people have Alzheimer's disease and 30% of our people had vascular disease, then 15% would have Alzheimer's and vascular disease. And that appears to pan out for vascular disease and Alzheimer's disease. Lewy body disease and Alzheimer's disease actually co-occur about twice as often as you would expect by chance alone. And I think that there is probably a relationship there that I don't fully understand at this point, but nature is probably trying to tell us something here. The flip side of this is that the risk for clinical dementia diagnosis is additive for these pathologies. The more pathologies you have, the increase your risk for dementing illness is. Okay, now I'm going to go into, this is sort of the didactic portion. I'm going to go into some of the finer pathology of how we diagnose Alzheimer's disease. And the characteristic findings are plaques and tangles, as well as amyloid angiopathy. Here are 3 different common stains that we have traditionally used to look at different pathologic features. The oldest one is the Bilshowski stain. Bilshowski is a silver impregnation method. It's basically the same chemistry as black and white photography. There are a lot of different silver stains out there that have been used for almost 200 years. And what they do is they tend to stain accumulations of protein black. The Bilshowski stain tends to have high affinity for 2 of the pathological features of Alzheimer's disease, the intraneuronal neurofibrillary tangles and neuritic plaques. Tau is one of the abnormal proteins that tends to be present in both of those lesions, although we tend to think of the plaques. Here's a plaque as being predominated by amyloid beta protein. They actually are compound lesions that also contain neuritic processes that contain a lot of the tau protein. Congo red is one of the non-specific amyloid stains. It stains any amyloidal accumulation of protein regardless of the type of protein. It happens to stain the deposition in vascular channels very nicely. I'm going to take a step back here and just pull up an image from a textbook that I wrote a chapter on, just to orient us to the cells that we're going to be looking at here. This is a photomicrograph at high power of gray matter from brain, containing a neuron. Here's the neuron. It's axonal processes, which are myelinated. There are small blood vessels, microglia, which are the resting monocyte population in the brain, and then astrocytes, which are the metabolic and structural support cells of the brain. And especially neurons and astrocytes are going to be important throughout the rest of the lecture. I can't talk about the pathology of Alzheimer's disease without talking just a tiny bit about the amyloid cascade hypothesis and how it, this is the dominant hypothesis for how Alzheimer's disease progresses. And there's a lot of support for this hypothesis. There are also some questions. Basically, neurons contain amyloid precursor protein, which is a single transmembrane protein in the endoplasmic reticulum of neurons that is normally cleaved by a enzyme group called alpha secretases that cleave it through a middle domain where it is broken down and removed from the brain. In the disease state, there are another two enzymatic complexes called the beta and gamma secretase, which instead cleave it at two different sites and produce this disease-associated protein fragment called amyloid beta. Amyloid beta aggregates. It's unclear whether or not it itself or small aggregated groups of it are somehow neurotoxic. These groups aggregate into amyloidal groups, which are beta-pleated sheet rich groups that are out in the parenchyma. They're extracellular aggregates. These are the plaques of Alzheimer's disease. It's unclear that these plaques themselves are, whether or not they are themselves toxic or whether they are somehow protective. Then there's a black box. It's not clear what happens at this point, but something happens to neurons where they begin to accumulate a different protein, microtubule-associated protein tau, dimers of which accumulate into fibrils within neurons, either causing or associated with neuronal dysfunction. This protein is present both in the prokaryon as well as out in the neuritic processes of those cells. And we end up with dead neurons full of these fibrillary groups of tau. This is the neurofibrillary tangle of Alzheimer's disease. These little neuritic processes also tend to be associated with these aggregates of amyloidal protein, and indeed it is the combined lesion where you have amyloid protein associated with neuritic processes that are associated with cognitive impairment. So a step back. So taking information from the population-based studies of aging, the National Institutes of Aging, along with the Alzheimer's study, proposed a study group to revisit the evaluation of Alzheimer's disease pathologically and clinically. This is back in 2012. This is a who's who of directors of the Alzheimer's centers. And they attempted to simplify and harmonize Alzheimer's diagnosis. They came up with a composite score that's been called the ABC score that basically took all of the previous methods for evaluating Alzheimer's disease and combined them pathologically. It was supposed to be agnostic to the methods for analysis, so people have used a variety of different staining protocols, silver stains, immunohistochemistry, amyloid stains to try to do Alzheimer's evaluation. This allowed people to use any of those techniques to try to create combined scores. Although I think that immunohistochemistry is, silver staining is the historically most common, but immunohistochemistry tends to be, I think, the most widely accepted. And they also included consideration of the non-AD pathologies, specifically vascular disease and Lewy body disease. So what is that ABC score composed of? And I'm going to start backwards and go CBA because those are the order in which these systems were developed. The first one that was developed was the CRAD score. So this is the late 80s, early 90s. CRAD stands for Centers to Establish a Registry for Alzheimer's Disease. And here they looked simply at cortical density of neuritic plaques. So these are those combined lesions that contain both amyloid protein and tau protein. And they asked pathologists who do not like to count objects under microscopes to just give a semi-quantitative assessment of how many of these plaques they saw in a couple of different regions of brain association cortex. And at this point, most people were still calling what we called late onset Alzheimer's disease simply senile dementia. And these were referred to by many people as senile plaques. And it was unclear whether or not these were a natural part of aging. I don't personally believe that. I think these are all disease associated. But they categorized these and then put them into age bins to try to decide whether or not they were really predictive of dementing illness. And it was difficult to get to Alzheimer's disease caused by dementia if you were above a certain age. The other unfortunate thing that they did here was they told people to look at section of the brain and find the single field that had the most pathology, and use that as their estimate. I can't think of a worse way to structure data than to say, I want you to look for the outliers in this data set, and those are, you're just going to keep the outliers and throw everything else away. We're not going to average it. As such, it's not surprising, really, that this system is not a very good predictor of cognitive performance. And it was supplanted in the mid-90s by Braque staging. Braque staging is based on the recognition that there appear to be progressive stages of Alzheimer's type pathology. Specifically in the deposition of these neurofibrillary tangles. Also, another thing that I think is of interest here, this is actually looking at individual damaged neurons involving cortical regions. If I had to hypothesize, what is a likely correlate of cognitive impairment, injury to neurons in cortex would be a pretty reasonable idea. This is a husband and wife pair, Braque and Braque, and they came up with a 7-level pathologic staging system, a 0 to 6, based on where those neurofibrillary tangles are, starting in the transinterrhinal cortex adjacent to the hippocampus, then involving progressive regions of the hippocampus, beginning with the C8-1 region, and then in the more advanced stages involving all regions of the hippocampus, as well as association and finally primary cortices. There are different ways that this has been realized in different Alzheimer's cohorts. The adult changes in thought study basically looked at the presence of neurofibrillary tangles in different cortical regions, looking at the highest level of pathology. So if they were present in the transinterrhinal cortex only, that was the lowest stage. If they were present in that hippocampal and in visual cortex, that was at the highest stage. Basically, though, a relatively small number of tangles out in visual cortex would put you to the highest stage, regardless of the density of tangles in other areas. The other studies used more sophisticated methodologies. The Ross-MAP study used a complicated averaging algorithm to try to produce a reproducible Braque score. Braque has been the mainstay of neuropathologic assessment. I think it's been the most consistent predictor of cognitive impairment and has been favored by neuropathologists throughout. The other interesting thing about the Braque staging is this is also where the idea of transsynaptic spread for Alzheimer's pathology became most prominent. The Braque's proposed a specific pathway mechanism that this pathology would spread from one region of from the transinterrhinal cortex throughout the hippocampus and out to cortex. bringing the hypothesis of the so-called prion-like spread of neurodegenerative disease, which I think is a good competing or concomitant hypothesis. The last part of that composite score is the A score. This is the tall phase. And I think this is an excellent example of expert consensus criteria sort of being driven by experts rather than good data. This extended the same idea of anatomic progression from the tau neurofibrillary tangles to amyloid, and looked at the regions where amyloid spread. Interestingly enough, it doesn't mirror the same pathologic spread as you see in the hippocampus. It doesn't go from sort of the mesial temporal lobe to the hippocampus to other regions of the brain. It has a very different pattern where the earliest location is in association cortex throughout the brain. So amyloid appearing out in the cortex early, then involving limbic structures, then involving deep nuclei, brainstem nuclei, and finally cerebellum. It's interesting that you have this order of progression, because it's actually not the same thing that you see in amyloid, in PET scanning for amyloid, which tends to appear in the deep gray matter nuclei first, and then out in cortex. This was included as part of the 2012 NIA Reagan criteria. There was a single paper on a small cohort that suggested this was a predictor of cognitive performance, and it made its way into those recommendations. I don't think there are many pathologists who like this. Number one, it changed our practice significantly and didn't offer us additional predictive information. Also, it took a nice two-dimensional grid where we could put plaques versus tangles and add an extra dimension. There could essentially be 27 different bends that we could put pathology into. Unfortunately, that tall staging doesn't really offer additional information to us. One of the things that the NIA Reagan criteria did was that they did attempt to unlink the clinical and pathological diagnoses. Their basic statement being that in individuals without cognitive impairment, if you had pathological changes, that's fine. This simply represents preclinical disease. But if you did have clinical impairment, well, that was probably a reasonable explanation for the dementia. If you look at the 2x2 grid, they also produced an estimate of the probability based on different pathological findings, whether or not they thought that the pathology would explain dementing illness by Alzheimer's disease. And you got a low, intermediate, or high criteria, or not. Of interest, there are some missing cells if your tall phase and your dendritic plaque phase don't match each other and the mismatch is too large. There's no place on this table for them. And in the real world, this actually ended up being a pretty common occurrence. It was very common to get a tall stage 1 and then have severe high neuritic plaque density and Alzheimer's pathology. Also, the only category that allowed you to say that something was high likelihood of Alzheimer's disease was high Brock stage with a high neuritic plaque score. Maybe that's reasonable. But it is not uncommon to see intermediate or low levels of neuritic plaques associated with high Brock score. And it's been unclear what to do with those. Initial evaluation of the Anaya-Reagan criteria mainly looked at inter-observer variability. And the other thing that this classification did is it rescaled those 3 different pathologies into just 3 levels. So instead of one being a 4 level, one being a 6 level, and one being a 7 level category, they basically did 0, mild, moderate, severe for each one. So you can get a score of A1, B1, C1, that's low TAL stage, low Brock stage, low CRAD stage. That did help with inter-observer variability. The kappa statistics up in the 70s and 80s that's excellent concordance between pathologist rating. Unfortunately, this doesn't tell us anything about how well this predicts cognitive impairment in individuals. This just tells us how well pathologists agree with one another. When these went out to look at individual, when these went out and were used to look at actual human cohorts, not surprisingly, that A score ended up being in no way predictive of cognitive impairment. And so I personally think that A score is not useful. Although you are likely to see it at least until the next level, the next evaluation of the Anaya-Reagan criteria, which should be in the next couple of years, I think. They also did recapitulate Lewy body disease as an important predictor of cognitive impairment. Also, hippocampal sclerosis showed up in actually several different cohorts now. And actually, as the adult, this is something that's been found in ROS-MAP. Age-related hippocampal sclerosis also tends to be a predictor of cognitive impairment. It's a couple of % of your dementing illness. And as it was statistically marginally significant in the adult changes in thought cohort, and as they have gained more numbers, it's now become significant. Okay, talk about Lewy body dementia pathology, just so that we're a little bit more, or so that we're reading comprehensive of the pathology seen. This is also a common reproducible pathology. About 10 to 13% of individuals in our cohorts have Lewy body pathology at the time of death. When it involves cortical structures, it's a strong predictor of dementing illness. It's generally associated with diffuse cortical atrophy. Here's a picture of what a Lewy body looks like out in brain. That's inside a neuron. It's a large aggregate of alpha-synuclein protein. It's a protein that's normally present out in the synapses. In the disease state, it tends to be centralized into the soma, where we can identify aggregates. It's pretty easy to find in pigmented structures like the substantia nigra. You don't have to use any additional special staining. But out in cortex, even histochemistry is very helpful to find it. It also has a progressive staging. It tends to appear first in the brain stem, and then ascends out into limbic and then other cortical structures. The last, and near and dear to my own heart, are microinfarcts. There are a number of competing ways to try to assess vascular disease in the human brain. This one system worked very well in the adult changes in thought study. Previously worked very well. Basically, these are the present of small infarcts identified microscopically alone. They tend to be between 50 and 500 microns in diameter. We only counted those ones that are old and therefore could be associated with a premortem diagnosis of dementia. We've been able to recapitulate this in multiple different cohorts. I'm not going to go too much into this slide. Suffice it to say that the number of ones that we catch on the slides that we look at is very small. And the actual number of these lesions may be much larger. And this is just saying that for about every one we find, we would expect that there are probably another 500 lesions that we do not find. What do I think these are? These tend to occur in the highest rates out in the cortical watersheds. And they are, I think they are evidence of a chronic low flow situation. I've looked at a number of different attempted animal models for these microinfarcts. The only one that really works, we tried microembolic models, and the one that looks similar, that appears to recapitulate the human disease are coarctation of the carotid model. So basically you put in a catheter into the carotid so that the flow is reduced. And this tends to produce little watershed infarcts that look just like microinfarcts. I think this is what's happening. They're predicted by blood pressure. These are associated with dementia. I was going to spend some time on my soapbox, but we're running slow. Basically, the take-home message from this was there's an awful lot of research that's been done suggesting that Alzheimer's disease was associated with dementia, with diabetes, that diabetes is a risk factor for Alzheimer's disease. And clinically, I think that's true. No pathologic study has supported that. Instead, it's all vascular disease, and I think that's not surprising mechanistically, but this is the number of papers that have been published based on the understanding that diabetes is a cause of Alzheimer's disease, and I don't think the human data actually supports this. That's just on that same thing. There was one major problem with these microscopic infarcts that's sort of plagued my life, and that was that they couldn't reproduce this in the Ross MAP cohort, in the Religious Order Study Memory and Aging Project cohort. And this was very troublesome to me because this is a group of studies that I think are the premier clinical neuropath population-based studies in existence. Right? Their pathology and clinical assessments are impeccable. And I was looking for the original paper that I was looking for the original source to demonstrate how there was this conflict. And they just published a paper, I just found this looking for this talk, where they went back and reanalyzed their data, and just as we found, what is important appears to be multiple infarcts that are occurring in cortical watersheds. Now that they're actually looking at this specific question, are these microinfarcts specifically in cortical watersheds? Are those predictive of dementia? And they are. It's very exciting for me. We're going to have to go back and attempt to reproduce this exact analysis. I actually got up at like 3 this morning to send some emails about studies I want to do. Okay, so now very quickly, I'm going to go into what we're missing. So I've talked about Alzheimer's disease, vascular disease, and Lewy body disease, and there is actually other pathology that we're missing. And part of that pathology is things that we have that I'm excluding because they're of lower prevalence. Now, something like frontotemporal dementia. I say lower prevalence because it's not 30% of the population. It's still an incredibly large group of patients. So I don't mean to, just because I'm not talking about them doesn't mean that they're not important. But I am going to talk about a little bit of an alphabet soup of acronyms that have appeared. And many of them are under this rubric of suspected non-Alzheimer's pathology, or SNAP. And this is basically the idea that we have, in biomarker studies, we have individuals that have evidence of neuropathology that is not specifically evidence of Alzheimer's disease. And so there are other pathologies involved. And as a neuropathologist, I find this a little bit, I'm uncomfortable with this, mainly because that's such, the N, the non-Alzheimer's pathology is such a broad, heterogeneous group, and I don't want to, I don't want people to become confused by this. I don't want it to be misled. This has been a concept that's been championed by a neuroradiologist male named Cliff Jack. Originally the AN concept, and then following that with the development of better biomarkers for tau, the now ATNs, so an amyloid beta group, a tau pathology group, and then a other neurodegeneration group. And I think I would be more comfortable with this idea if the N also had maybe a separate vascular disease group, because I think that the mechanism underlying that group is probably different. So just ATN to me is a little bit too, it's too large a bin to use. But this is another consensus. This is another consensus idea coming out of the NIA Reagan group. This is just my idea of what these individual bins probably actually mean. And well, I guess I'm over time, so I won't talk about some of the other, but there are other types of dementing injury that are probably not being accounted for by the previous system. These include primary age-related tauopathies. So this is tau disease that is associated with dementing illness that is not amyloid. Aging-related tau astrogliopathy. This is tau pathology that's in astrocytes that's also incredibly common, and it's unclear whether or not it's a predictor of dementing illness. And it's a complex pathologic thing to deal with. There's an awful lot of different ways you can look at different astrocytes and try to bin them into groups, and I'm not sure that I believe any of this. And finally, hippocampal sclerosis is now, at least in the neuropathology world, getting an update as cerebral age-related TDP-43 pathology with sclerosis, based on the finding that the vast majority of individuals that have hippocampal sclerosis also have mislocalization of this TAR DNA binding protein, which is an interesting protein. It is present in a large group of frontotemporal dementias, as well as here. The only thing that I would say here is that it's a finding and not a diagnosis. TDP-43, it's found in different contexts. It's unclear what it actually means. But it is, as the prevalence of new cases of Alzheimer's appears to decrease with age, this hippocampal sclerosis, new hippocampal sclerosis surrogate appears to increase with age. So it's a late age cause of dementia. So I'll open it up to any questions. I apologize for running over.
Thank you very much for this very nice overview. So we're a little bit late, but maybe one or two questions.
Yes, ma'am.
Really interesting
In these cohorts, we tend to see a relatively small number of those cases, and so we haven't done analysis based on them. But I think that they're interesting. I mean, I think that they are interesting diseases, and I'm interested in other specific cohorts associated with them.
Brief comment about in animal model. Is there a fundamental reason why the animal models don't perfectly reproduce Alzheimer's, or do they?
No, they don't. I think that at least in I think probably, at least in mice, they simply don't live long enough. I don't think that they have the lifespan required to produce these pathologies. Neurofibrillary tangles appear to take a decade to form. So I think that's what's going on.
Thank you very much.
Thank you.